All right, everyone, let's get started. Okay, can everyone hear me? And can everyone see slides? Okay, great. Okay. So, general housekeeping for today. We're working on chapter 36 today. You should have had your pre-lecture assignment completed last night. Thursday, we're going to be, again, out of order in terms of the chapter numbers. We're going back to chapter 28, and we're going to finish up my time with you with fungi. You have an assignment due Wednesday night, okay? Now, office hours. So, sorry. Ooh, that's a typo in there. No, not after today, after Thursday. So, Thursday is my last lecture with you. And given that I'm not going to regularly be lecturing and looking at how many people are, or rather are not, coming to office hours, so I'm not going to regularly hold office hours after this week. I will, of course, be available to all of you. Just email me and we can find a time that works for you, and then we can meet. Specifically, the reason that I am doing this is because some of the chapters that we've covered are going to be on your next midterm exam 2, specifically chapters 26, 27, 36, and 28. They're there on the slide. Those are going to be covered along with other new material that Dr. Arnott's going to be discussing with you. That's going to be covered on midterm 2. So, of course, I am available to go over any and or all of that should you desire. Okay. That being said, I have some good news for those of you who were very enthusiastic about Dr. Arnott taking over. So Dr. Arnott is starting his lectures next Tuesday, and he will be lecturing for the remainder of the course. Okay, so expect to see a different face and a different voice come Tuesday. Okay, any questions about any of the housekeeping stuff? There is a date for the next midterm. It's on your syllabus. That's correct. And that is going to remain the same date. Okay. Any other questions before we go on? Okay. All right. So today we're talking about reproduction in flowering plants. I have here an image that actually comes from sort of an amalgamated source that has looked at the tree of life for plants. And as I mentioned when we started talking about plants, angiosperms, the flowering plants, have really done an amazing job in diversifying. There are so many different taxa of flowering plants. Here it just helps to kind of show you some more of that. Okay, so you see species richness for these different taxa that is color coded. This is just angiosperms, and this is really isn't just a very, it's not down to the species level, but yeah, this should help you see sort of the diversification that has happened in the angiosperms. Being able to flower once you had land animals, in essence pollinators, has really helped angiosperms diversify, okay? Being able to attract those pollinators. So that's something that I want you to keep in mind. Okay, so key concepts for today. We have just 3. So we're going to go into a little bit of depth with them, right? So let's get started. Most angiosperms reproduce sexually, okay? I mean, there's nothing really super surprising here, right? You've learned about flowers and that they are the reproductive organs of plants for some time, right? Now, I want you to kind of recall back to some of our earlier lectures where I talked about how sexual reproduction is a mechanism for increasing genetic diversity, increasing and maintaining genetic diversity, okay? So by reproducing sexually, plants are protecting and trying to expand their genetic diversity. And it's that genetic diversity that is important when we talk about evolution, okay? So what does it look like in plants, sexual reproduction? Well, we've got mitosis involved, we've got meiosis involved, and we've already talked about this alternating haploid and diploid generations, okay? So it is, though, different than it is in animals, right? So in animals, there's no multicellular haploid stage. In animals, you have haploid cells that are then used for reproduction, and that's pretty much it. But in plants, there are multicellular stages in both the haploid and the diploid stages. So it's important to realize those differences, okay? So if we compare sexual reproduction in animals to plants, in animals, those germline cells are determined before animals are even born, right? So those germline cells are there. In plants, particularly the angiosperms, right? Cells form these gametes that are determined only in the adult stage, right? So basically, if you're a plant, you're not really doing anything biologically related to reproduction until you're an adult, okay? And a lot of this really depends upon the environmental conditions that you find yourself in when you are an adult. Now, I kind of want to call your attention back to something else that we talked about earlier in the semester, right? Remember how in prokaryotes really are reacting to the environmental signals that they're receiving. And I said that eukaryotes really have a lot of internal signals. Plants do both. They have a lot of internal signals, but they're really depending for reproductive purposes in particular. They're depending upon environmental signals as well, okay? All right, in animals, you have meiosis, and that directly produces the gametes. Not so in plants. In plants, meiosis produces spores. It's mitosis that produces the gametes, okay? So again, differences, some of the major biological differences between animals and plants, okay? And just to kind of reiterate how it works, the life cycle in plants, but I'll let you guys go through that on your own time. Okay, now, I also wanted to talk about the layout of flowers. Flowers follow a pretty basic layout. And this is actually one of the things that I think your textbook does really, really well, is show this, and in particular the color coding, which they carry through the whole chapter, and I'll try and highlight for you as well, okay? So flowers have 4 concentric groups of tissue, okay? All of these 4 different groups are basically modified leaves. They evolved out of leaves. They are specialized leaves, okay? So the one in the center is the carpel. This is your female sexual organ, and that's what's going to contain those female gametophytes, okay? Then you have the next ring out here in green, are the stamens. The stamens are the male sexual organs. That's where the male gametophytes develop. That's where your pollen develops, okay? Then you have the petals, which is probably the most easily identifiable sort of feature of flowers, right? I mean, everybody knows the petals of a flower. That's where you get all these amazing colors, shapes, things like that. And then the outermost ring, that's your sepals. These are all modified, evolved from leaves of the plant. Okay. Now, I've mentioned before perfect and imperfect flowers. Most angiosperms have perfect flowers. What do we mean by perfect flowers? They have both the male and the female sexually reproducing tissue, right? They are both male and female. Imperfect flowers have only one, either a male flower or a female flower. Male flowers have the stamens, female flowers would have the carpels, okay? Now, also with imperfect flower, you have to sort of denote differences in the plant itself. Is the plant monoecious or is it dioecious? Okay, what do we mean by that? Monoecious have both male and female flowers. Remember, these are imperfect now, right? So you have male flowers, you have female flowers. So monoecious have both male flowers and female flowers on the same plant, okay? Dioecious, you have the whole plant is either male. In other words, it just has male flowers. The whole plant just has male flowers. Or the whole plant just has female flowers, okay? Again, very, very different approaches to how sexual reproduction happens in animals. Okay, so with that, let's go to our iClicker question. Okay, right, so why might natural selection favor angiosperms evolving perfect flowers, but separate sexes in most animal species? So why these two different strategies between angiosperms and most animals? I don't see a typo on the slide, so person who had that concern. Sessile, that's a good question. Sessile means that you can't move. You are in one place. Immobile, exactly, Isaac. Thank you. Just a couple more seconds. Okay, let's see what we've got here. All right, great job, guys. Most of you selected the correct answer. 78% of you selected all of the above. That's exactly right. And when we look at it, we can see why this is the case. Okay. Yeah, plants are sessile, right? So how you're going to reproduce becomes a really important question, right? So having perfect flowers where your male and female organs are out there, that really helps. Plants rely on passive reproductive methods, right? You can't do anything to promote, you can't actively do anything, right, in terms of actions, moving, or things like that to promote reproduction. So you have to have these other ways of reproducing, passive ways. So you know, and this again shows why angiosperms have become so successful, because they're very, very good at basically convincing an animal to help them pollinate. They're pollinators, attracting those pollinators and getting those pollinators to then visit others of their species and thereby helping them to reproduce. Animals, on the other hand, can actively seek out mates, right? There are many animals that there is a specific part of their life cycle or their year where that's what they are doing. They will forego concerns about survival in order to reproduce. Think about salmon. Salmon stop eating you know, to travel back upriver to lay their eggs. You can think about other animals that may actually engage in competition, physical battle. Typically, the males will do this physical battle to win access to mates. Okay, animals cannot devote the same amount of resources to reproduction. Yeah, right. That's true because you don't have to have so much energy that gets devoted to moving around, searching for food, you know, all these different things. That is a function of where you land in the environment. If you're a plant, you don't have to worry about those things. You can put more resources into trying to reproduce. So that's why it's all of the above. Well done, guys. And I lost the share. No, there we go. I got it back. Okay, great. Okay, so let's talk about some of these imperfect, and monoecious and dioecious. Okay, this becomes really important, particularly in agriculture. So we're going to talk about an incredibly important agricultural plant, corn. Okay, so here are corn plant. You see some corn plants here. Corn is imperfect monoecious. Okay, in other words, flowers are either male or female, and every corn plant has both male and female flowers. And here you are. I'm showing you those flowers. So at the top of a corn plant, that is the part that is male. Those are stamens. Okay, the pollen develops here, and then you can see that corn is often air pollinated, wind pollinated, right? So those stamens, they're way up high so that the pollen can be carried away by the wind more easily. Okay, and then here's the flower, the female flower. Okay, right there. So all that corn silk, right? Those are the carpels. Okay, corn is an imperfect monoecious plant. Okay, and it has the male and female flowers. The female flower obviously is what develops into the fruit that we harvest, either for us to eat or other animals to eat, or to make other products that we then eat. Okay, and this is incredibly important because of a thing called tasseling. Now, many of you may never have heard of tasseling or detasseling, but this is something that is actually really important, particularly for people about your age in many parts of the world where they grow corn. There is many teenagers, young people have summer jobs that are detasseling. So here's what we mean. When you want to produce corn seed, so you're not growing the corn to eat or to feed to another animal. You're growing the corn because your crop is seed for next year's corn, right? So you sell the seed. Seed companies do a lot of this, okay? Now, you don't want, because of genetic diversity concerns, you don't want the corn to self-pollinate. You don't want pollen from the same plant to pollinate the flowers on that same plant. You want to maintain genetic diversity, right? So you want pollen from other plants to pollinate the corn plants and produce corn seeds. Okay, so what you do is typically they do it in rows. So one row you plant corn and you want that to be your male flowers. And then the next row you plant corn in a field and you want that to be your female flowers. And you alternate male, female, male, female, male, female. And then basically what you do is the ones that are going to be female flowers, you don't want pollen from them. Okay, so you have to have people physically go down the whole row and take off the stamens. Basically, remove all of this from the top of these plants. Now, if you've not seen adult corn, corn can grow probably about 6 to 7 feet tall, so taller than the vast majority of humans. Okay, and there's a lot of corn in the cornfield, and so you would have a summer job where you basically go over typically like a 2-week period into a field, and your job is to take off every other row, take off all those stamens. And I've never done it. I've spoken to people who have, and I've heard that it really is very difficult. First of all, you're in heat. You have your hands above your head for most of the day. And if you don't think that's a big deal, I invite you to sit there for 10 minutes with your hands, holding your hands above your head, and let me know how you feel after that. And that apparently the corn plants are really pretty tough. So think about if you've ever taken a husk off of a corn cob, how tough and fibrous that stuff is. Well, that pretty much is the corn plant. And so it's very damaging to the hands to have to do that. Okay, so that's corn detasseling. Now let's talk about an imperfect dioecious plant. This is Ilex opaca, or American holly. And basically, you have American holly plants that are either the whole plant is male or the whole plant is female, okay? And you tell when it flowers. So here are the flowers. They're very small flowers. And here is the female flower versus the male flower. And you can see on the female flower, it looks like it has stamens, right? But they're not. Those are faux stamens. They're not really stamens. Those are basically just funny shaped, for lack of a better way of saying it, funny shaped petals. Remember that stamens and petals are both modified leaves. They evolved from leaves. So basically here, it is a stamen that is shaped, a stamen-shaped petal, okay? As opposed to the male flowers where you can see, yeah, that's definitely a stamen. You can see pollen grains up here as well, that golden sort of color. And you can see that the carpel itself in the center, or where the carpel would be, is not developed, right? So that would be the male flowers, okay? All right. So the megagometophytes, right, which are the female ovules, ovary, right, the embryo sacs, and the microgametophytes, which are those pollen grains, okay, they develop on different parts of the flower. And again, these are highly specialized, right? And this is where you get that development of these sexual cells, right? And you can see here, as a matter of fact, particularly for the pollen, where meiosis happens. Here, meiosis happens right here, but then after meiosis, again, you have mitosis. Very different than in animals, where you have these cells and they undergo meiosis, and boom, those are your gametes, okay? Not so with plants. Okay, so let's talk about self-pollination. There are certain species that depend on self-pollination. Some species of peas do this, where they actually self-pollinate before the flower even opens, okay? Some species of figs, particularly from Africa, also do this, okay? This leads to a lot of homozygosity, right? In other words, the reduction of genetic diversity. But most plants actually have several different types of mechanisms whereby you're not to guard against self-pollination. Okay, you can have the physical separation of the male and female gametophytes, right? So we've seen this. We've talked about this, where you can have in the monoecious plants separation of male and female flowers, right? That's going to reduce the chances of self-pollination. You can also have where the male and female flowers on the plant will actually mature at different times. Okay. You can have these mechanisms working on the genetic level, where some plants are what's called self-incompatible. They reject their own pollen. Okay, if their own pollen lands on their own stigma, there is a genetic mechanism where where they get to reject it. And basically, this most often is connected to what's called the S locus. So the S locus is this particular place in the plant's genes where there are many alleles, right? Many different versions of what that gene actually is. And if it is the same, if the female part of the plant senses that the S locus on the pollen is the same as itself, it rejects that pollen. Okay, so it's only looking for something different. Okay. So this is really interesting, and you can see why it's important for there to be many alleles. If there were just two alleles, right, then you would be rejecting half of all pollen that would land on a particular carpel. Okay, so not what you want to do. So yeah, okay. Plants, when they do fertilize, actually use a system of double fertilization. Okay. Um, Double fertilization, where one sperm cell is going to fuse with the egg cell, fertilization as we understand it, right? And that forms the diploid zygote. The other sperm, though, is going to form with fuse, excuse me, with these polar bodies, basically these polar nuclei in the central cell, and that actually ends up forming a triploid or 3N cell. In other words, the number of chromosomes there is going to be incorrect for the plant. Okay, and that's fine. Because of what ends up happening with those cells. Basically, you have the zygote, right? It starts undergoing mitotic division and starts basically growing into the embryo. But that triploid nucleus, that's going to undergo mitosis, and that ends up forming your endosperm. And remember, the endosperm is the nutritional source for the seedling in the seed. Okay, it gets digested by the developing embryo. Okay, so now before we go on, actually, let me stop. I just realized I didn't put in a question slide, so I would like to stop here for questions. So, all right, shoot. Let's see if we can tackle them. Melissa, go ahead.
Yeah, so in one of the earlier slides, there was a picture of a flower showing the stamen and the carpel. I think it was of a lily. And I've noticed that like those flowers, like the carpel part is like a little gooey. I was wondering, is that to like attract like pollen and to keep hold of it?
Yeah, it's exactly right. So that pollen that might come in to that stigma is going to be able to stick in essence, and if it matches, if the S locus isn't an issue, or other things like that, that pollen can then pollinate that plant. That's exactly right, Melissa. Dana, why don't you ask your question?
Hi, Professor. My question actually kind of builds on Melissa's question. So I was wondering if the fact that the female plants have that stamen-looking structure, was that due to evolution, or was that kind of a sexual adaptation to attract pollinators to bring pollen to the female?
Yeah, so the pollinators aren't going to be attracted. Most pollinators are not attracted to shape, right? Most of the pollinators are attracted to something else, whether it be the look or the smell or the color or something like, well, I guess color kind of backs up, going with what you were suggesting. No, I think it probably just has to do with, as that female flower is developing, there are certain plant hormones that are going to be there that are going to be in both the male and the female, and that's how it affects the development of those particular petal structures. So, so it's not going to look exactly like that in every species, but you may see. And you saw it, by the way, in the male flower as well. You had the big stamens that were standing up there with all the pollen up at the top, right? And you could still see in the center that that greenish colored stigma, basically, right? Where the female part would have been if it were a female flower, but it was also underdeveloped. So in a lot of ways, it's just basically that the male and the female organs are developed or not developed, usually because of internal hormone kind of signals. Okay, is that helpful?
Yes, thank you, Professor.
Yep, you got it. Any other questions? I'm just trying to peek at some of the questions in the chat. Do you think one day it'll become dioecious because of human involvement? Well, certainly. I think that, by the way, for all agricultural products, if it weren't for humans, they would change rather rapidly. Okay, think about fruit. Wild apples are not the size of the apples that you buy at the farm stand or the market. Okay, these apples are tremendous compared to wild apples, which tend to be much smaller. Think of like a crab apple, kind of a thing. So it's humans and the artificial selection that we've imposed, as opposed to natural selection, that has selected for these particular varieties. Bananas too. Bananas are a great example. Natural bananas, so much smaller than agricultural bananas. Okay. Okay, so Dana, I think we kind of addressed that question. The purpose and advantages of double fertilization, particularly the triploid cell formed. So Nathan, yeah, basically Nathan, that triploid is just how it happened in an evolutionary sort of way, but that ended up becoming the endosperm, becoming the food source, so that embryo in the seed can survive for long periods of time. Okay, so I hope that helps sort of clarify. So how plants attract pollinators, many different ways, right? So we mentioned some of them. Scent is one of them. The way that they look, the way that they smell, these different things. It can be there. It's really interesting. There is a species of orchid that actually orchid. I believe it's an orchid. It may not be an orchid, but anyway, where it actually, one of the petals looks like a female queen bee. That is searching basically at the stage of life where it would be searching for a mate. So male bees land on this petal and basically try and mate with it. And in so doing, they are picking up pollen from the flower, and then they realize, yeah, this isn't working out the way that I thought it was. And they leave, they search for another flower, and they say, wait a minute, there looks like an opportunity for mating right there. And they go there and they carry the pollen from flower 1 to flower 2. So there are lots of strategies that plants use to attract their pollinators. Color, remember I said that color is important for bird pollinated species. That's why a lot of the hummingbird attracting flowers are red. Okay, many species of hummingbird like that color red. That's why many fruits are colored red. Birds seem to be attracted to the color red as a signal of ripeness. That's why many berries are red. White, I think I mentioned that bat pollinated species, the flowers are almost always white because bats, it's easier to see in the dark. And of course, bats with their vision, some famous examples that because vision is not their primary sensing organ, it's sonar, for how they search out food. So, but being able to see those flowers at night is beneficial. Okay. Which of the two, perfect or imperfect, have more genetic diversity? That actually would make a great eye clicker question. Sorry, I didn't think of it, Yvette. But I'm going to leave that there for all of you guys to think about, and we'll move on. From there. That's a really good question. I'd love to talk about it too if we have some time, maybe at the end. Okay, but I do think we probably need to move on. Okay, so let's talk about after fertilization, right? We get seed and fruit. So fertilization happens, right? Now we're starting to grow the embryo, the endosperm, and these integuments and carpel. What the heck are these integuments that you're talking about? Integuments are the tissue that are surrounding the ovule. It becomes the seed coat. So integument, tend to be kind of tough. Think about the outside of seeds, right? You get that seed coat. It has to be protective. It has to be pretty tough. The carpel becomes the wall of the fruit that surrounds the seed. Now, as seeds develop, obviously they don't want to be heavy for many reasons. Water adds weight, so they're going to, in a lot of ways, dehydrate. You also don't, when you have dehydration, that helps promote the dormant phase that a seed needs to go through. Ovaries and seeds can develop into fruits. The fruit has many different purposes. One of them is to protect the seed from damage, infection, particularly by things like a fungus, which we'll talk about on Thursday, and in a lot of ways helps with seed dispersal, right? So think about strawberry. Every strawberry that you eat, you don't pick off all the seeds, right, that are on the outside. You just eat those seeds. And those seeds are much tougher. They're going to be able to pass through the digestive tract and be deposited then when an animal defecates. Some species produce fleshy edible fruits. Some fruits are dry and inedible. Okay, so there's really a range depending on the mechanism that natural selection has selected for over time for that particular plant. Okay. Did I lose? I lost the share. No, there it is. Okay, great. So let's take a look at some examples of seeds that you would be, and fruits that you would definitely be familiar with. Garden peas, right? So the pea pod is the fruit, and each individual pea in the pea pod are individual seeds. Tomatoes, you've got the tomato fruit. And then you can slice it open and you can see all the individual seeds, okay? All the individual seeds inside that fruit. Corn, so here is an image of a corn kernel, and you can see you've got the embryo, okay? And we just basically eat the whole fruit. Now, speaking of how tough that integument can be, I'm sure that particularly young children are amazed at what happens when you eat a lot of corn, and then that corn passes through your digestive tract, and the seed coat, that integument, does not break down, and it just gets passed through the whole digestive tract as a whole, not broken down. Okay, so seed dispersal. If you don't have mechanisms for seed dispersal, basically the seed is just going to fall to the ground near the parent plant. Now, this has some benefits. and it has some drawbacks. Its parent, right? That was a successful environment for the parent, so it's likely to be successful for the next generation as well. You just might have been in a good spot. But unfortunately, if you are the parent plant and your offspring is growing right next to you, the two of you are now competing for resources, water, nutrients in the soil, sunlight, right? So you're going to be competing directly with your offspring, which from an evolutionary point of view, not the wisest strategy. Okay, so being able to disperse seeds over a wider area is really beneficial. It also helps with things like genetic diversity. However, you have to realize that not all those seeds that are dispersing are going to find favorable environments. So you may need to produce more seed to end up with the same number of viable plants in the next generation that are your descendants. That's why some fruits have wings, right? And I think I mentioned last time the helicoptering of those maple leaves, right? Some of them can be very feathery. I think it was actually one of you guys, and I'm sorry, I forget who it was, who mentioned like dandelions and things like that, and how they're adapted to wind dispersal. And of course, many fruits hitch rides on or in animals, right? So we've already talked about how you can hitch a ride in an animal if that animal eats the seed, ingests the seed. Can't say eats because the seed usually passes through unharmed. But also on those burrs, right? You can have sticky sort of seeds that grab on, might be sort of Velcro-like if you think about burdocks or things like that, that stick onto an animal and then will fall off in a different place at a different time. We'd mentioned already last time, but this is a great time to bring up again that water can carry seeds away. Coconuts are a great example. Coconuts can float for thousands of miles between islands in the tropics, end up on a beach someplace else. The coconut is very well protected from being, it's not affected by being in salt water for long periods of time. It ends up on a beach someplace else and it can then germinate. So it's well known that coconuts are able to do this, okay? And again, seeds and fruits being able to pass through animals' digestive tracts, okay? Okay, so let's talk about some of those hormones. I already mentioned some of those plant hormones, so let's talk about them. Abscisic acid, or ABA, okay? This is one of the main hormones that controls development of seeds in plants. During early seed development, abscisic acid is going to be low, but it increases as the seed matures. So you can age a seed by taking measurements of abscisic acid. Abscisic acid stimulates the endosperm, remember that's that 3N part of the plant, to synthesize these proteins that help with storage and basically protect that embryo over a long period of time. Abscisic acid also, importantly, stops seeds from germinating too early. And I mean really early, like while they're still on the parent plant. And that would be what's called viviparity, okay? Viviparous species are species that give birth to living young. We are a viviparous species. Humans give birth to living young. If you're not viviparous, you can be oviparous, where you produce sort of an egg-like structure, or there's ovoviviparous, where you produce the egg-like structure, but that egg-like structure then develops internally. Some fish are ovoviviparous. Okay. Okay. So let's see. Seedlings are unlikely to survive and establish and cannot establish. Okay. So talking about when you get a seed that germinates too early, it hasn't even left the parent plant yet. If it germinates that early, it's extremely unlikely that it's going to survive, right? It's germinated in the wrong place. The resources are not there for it to develop into an adult plant and end up making seed of its own. We also, for agricultural purposes, really want to control this, because if the seed germinates too early, it's going to destroy whatever that agricultural product is. Sometimes it's the seed, sometimes it's something else. So for instance, in crops such as wheat, the grain is damaged if it started to sprout, right? Why? Because think about what it is that we want from wheat. Right? We are after the seeds, particularly the endosperm part of that plant that holds a lot of that energy and that nutritional value. Abscisic acid plays a role in maintaining seed dormancy. I think obviously that's pretty obvious from everything else in the slide. Okay. All right. Now let's talk about different kinds of plants. This goes along with hormones, because when hormone levels rise, that's going to control reproductive processes. So does this need to happen very quickly? It happen over a longer period of time, and we divide plants up basically in how long the life cycle takes. If you've done any gardening, or you have family members that are gardeners, they'll know this well, right? So you can have annual plants. Annual plants go through the whole life cycle in 1 year, 1 season. Okay, they don't survive to the next year. After flowering, so they're going to flower quickly. After flowering, most of their energy is going to go into seed, sort of seed production, seed development, fruit development, and then after that, the plant dies. It's not going to come back the next year. Annuals survive for 1 year. Then you have biennials. Biennials actually take 2 years to go through the entire life cycle. They have a vegetative first year, basically where they concentrate. So remember when we talked about evolution, right? And we said that there are 2 components to fitness. There's surviving and there's reproducing. So basically biennials, what they're doing is they're concentrating on different things in each year. The first year, it's about surviving. So you want to accumulate and store energy sort of resources, carbohydrates basically, right? And you can store them in your roots, or you can store them in your stems. We know a lot of these plants, even though you may not realize it. So for instance, carrots and celery, 2 vegetables, right, that are very common in most households. Those are biennials. We harvest them for food in their first year, not their second year. In the second year, they're concentrating on reproducing. But in their first year, they're bulking up, in essence, accumulating energy and storing it in the root, carrot, or in the stems, celery. Okay, and that's just 2 examples there. In the second year, they're going to then use that energy that they've accumulated and really go all out for reproduction. Okay, then you have perennials. Perennials live for some number of years, and their life cycles and what they do in 1 year versus 2 or longer, really varies. Okay, they may flower every year, but then after flowering, they'll go on, and they'll go on and grow again in years to come, in seasons to come. Some species, they have a reproductive cycle every year. Many trees do this, right? So I give the example here of the maple trees every year, or oak trees. Every year, think about an oak tree seed, its fruit is the acorn. That oak tree is going to produce acorns every single year. Other trees produce every other year. If you like Honeycrisp apples, for instance, Honeycrisp apples, I believe, go on a biannual cycle where they produce apples every other year. Okay, then you have other plants that grow for many years. Then they flower once, okay? And then once they're done flowering and reproducing, they die. Okay, so you get a lot of diversity in the strategies, the reproductive strategies there. So olive trees. Okay, see, there you go. This is another great example. Thank you. Okay, so determining growth and flowering, right? Okay, so you have the apical meristems. So you have the apical meristems, right? That's the part of the plant that's growing. They're going to continually produce leaves, stems, buds, things like that, okay? But if it becomes what's called an inflorescence meristem, it produces a bract, and that new meristem grows off at an angle. You guys have all seen this, and I'll highlight it in a minute, and I think it will make more sense. Just bear with me for a second, okay? That new meristem can become a floral meristem, which leads to a flower. Again, you guys have all seen this. Just bear with me, okay? Floral meristems produce 4 whorls of organs: the sepals, the petals, the stamens, and the carpels, okay, which form short internodes, which determine growth. Those 4 whorls, you've seen those before as well, okay? So hang on, bear with me. Here's a plant, okay? And you can see the meristem up at the top, okay? Now, if that meristem is stimulated by specific plant hormones, right, it can form an inflorescence meristem, right? So as the plant grows, that becomes a flower. Or it can have a situation where this becomes a bract, another branch basically that goes off that forms a flower itself. If you have seen rose plants do this all the time. So if you look at a stem on a rose plant, you can see these meristems, right? These floral or inflorescence meristems, right where there'll be sort of a little leaf. You can see that, and that has the potential to grow into another bract. Okay, I suggest you take a look next time you see some of these plants. You'll notice this happening as well, not just on roses, many different plants. Okay, so as I mentioned, flowers evolutionarily come from leaves, okay? And there are sets of genes called identity genes or organ identity genes. They work together. They're labeled A, B, and C. That they are non-coding genes. Remember what non-coding was? They don't code for a specific protein, but they may control what proteins do and don't get translated. So these are sort of, these can be switches or promoters or things like that, right? So you have 3 of these, A, B, and C, that encode transcription factors that act in combination to specify the type of organ produced. What the heck does that mean? Basically, what or when these genes are expressed, that determines what the outcome of that cell, that tissue, is going to be. Okay, I think this figure helps identify. So you can have, here are those genes, A, B, and C, color coded here. And you'll notice that A and B will combine and will attach on the DNA to a promoter that will basically switch on this gene, like kick it into high gear. And that gene promotes, not promotes, but that gene gets translated, and it creates proteins that then cause that cell to develop into petal tissue. Okay, and again, here you'll notice, by the way, there are those whorls, and one of the things your book does really well is the color coding, okay? And you can see how the color coding remains through all of these, and you can see how specific tissue, specific cells, very early on in the meristem, different genes are getting switched on and switched off, controlled by these floral identity genes right here, okay? To say, okay, this bit of tissue here is going to, in green, that's going to become your stamens. The bit of tissue in the center, that's going to become your carpel, okay? The tissue on the outside, that's going to become your petals and your sepals, okay? So that's all controlled by combinations of these genes, these floral organ identity genes. So when is the next question? When do these things happen? When does a plant flower? How does the plant know to flower? A lot of this has to do with available light, okay? So you have short day plants and long day plants. Short day plants, they flower when the day is shorter. Now, interestingly, they flower when the day is shorter after longer days. So in other words, you've already had the longest day, and by day I mean amount of daylight, right? And now the amount of daylight is getting shorter and shorter and shorter. That becomes the environmental trigger to signal it's time to flower for short day plants. This happens after the summer solstice, which is the longest amount of daylight, right? And happens in late June. So this is going to be the flowers that, the plants that flower in late summer. And I give you some examples there. Coffee does this, morning glory, poinsettias, things like that, okay? Then you have the long day plants. They flower when the day is longer than some critical value for that plant, okay? And that's going to be midsummer, basically late June, end of June, beginning of July, okay? And interestingly, we talk about the amount of light, but it's not the amount of light that is the actual trigger. It's the amount of dark, okay? Night is the actual trigger, not light, okay? So you can actually foul up some of these plants if you sort of did this inside as opposed to outside, and you were the one controlling the amount of light. And if you had increasing amount of light each day, and then you gave it all of a sudden a huge shift in the amount of dark, and then go back sort of to the same pattern of longer and longer light, you could completely foul up the timing of some of these plants. Okay, this is also more prominent in plants that are not tropical, right? So remember, tropical plants actually experience much smaller shifts in the amount of daylight and dark each day. Okay, all right. So now we've basically have talked up until this point only about sexual reproduction in plants, but as it turns out, plants can also reproduce asexually a few different ways, and we're gonna talk about that. So here's an example: iris plants. Irises are common in gardens around the world. I have some in mine. So they produce both. They can produce asexually. They also produce sexually, but let's talk about the asexual part. When you reproduce asexually, you're producing a clone, a genetically identical offspring. Okay, so genetic diversity, right? You're not getting a lot of genetic diversity when you reproduce asexually. What's good about it is that if the parent plant is done well, right? and I mentioned this sort of strategy before, if the parent plant is done well and you can create another offspring that is genetically identical to you, so it's going to theoretically at least grow the same way. It's got all the same genes as you, same alleles as you, and it's in a very similar environment, and that environment has been good for you, it's going to probably be good for that offspring. Okay. Um, so it's a good way for preserving successful genotypes. Uh, for plants like irises, they actually have what's called a rhizome. The reproduction happens at the root level. So, for instance, if you plant one iris, and it's a good spot for irises, and you come back in a couple of years, you're going to notice that all of a sudden you have several irises in that same area. Those are clones of each other. The rhizome has it's reproduced asexually through the rhizome, and the rhizome is spread, and many plants growing there. Okay, then you can have asexual reproduction on many agriculturally important um, plants. For instance, strawberries do this, right? So you can, and you'll see this if you ever see strawberry plants, you'll see these they look sort of like horizontal stems. They're called stolons, okay? And basically the plant will form this horizontal stem, and it'll go over to a different spot, and another plant will grow, but that plant is a clone. Interestingly enough, almost all strawberries sold commercially are clones. Okay, so if you did genetic testing, you're going to find very little genetic diversity on, particularly in the U.S., on strawberries sold in markets that are not grown necessarily. But even the plants themselves, almost all the plants, strawberry plants in the United States, come from just a few producers in California, and they're all clones of each other. So, so there you go. Okay. You can also have the shoot tips. So basically the plant is growing, but it'll sag, and it sags, and it sort of reaches the ground, and then all of a sudden it'll grab the ground, form some roots, and form another plant. Blackberries do this. Potatoes, I'm sure that you guys are familiar with those eyes that grow on a potato. If you take a potato, and you basically stick it in a drawer or something like that for some period of time, not even that long, a couple weeks, right? It'll start to develop those eyes, those protuberances that grow on it. That is basically asexual reproduction in the potato, and that would grow into another plant. If it has the opportunity. And I already spoke about rhizomes, these stems that grow underground. Bamboo is really good at this. As a matter of fact, bamboo can easily become an invasive plant, a destructive invasive plant. If you plant it, these rhizomes just spread everywhere, and all of a sudden bamboo is everywhere, and it's very, very difficult to remove from an environment once it's invaded. Along the same lines, you can have bulbs and what's called corms. Bulbs and corms, they're basically underground modified stems. Bulbs are very fleshy, right? A lot of tissue in them. They are really good at storing energy. And storing it underground, and they can each develop to new plants. Some examples: lilies do this. Onions are a bulb. Okay, so you're actually eating the bulb. Garlic is a bulb. Garlic's really interesting because each one of those cloves is a bulb, right? So you can, if you wanted to, you could take a clove of garlic, plant it, and it would grow an entirely new plant. Okay, corms are very much like bulbs, but they're really more modified stems. They're not as fleshy. Crocuses, which are blooming right now. Crocuses are wonderful, tiny little flowers, lots of different colors. They are among, if not the first, among the first plants to bloom every spring. It's always nice to see them starting to bloom. They're blooming now. It's a signal that spring is not far away. Crocuses are also important for agriculture. It is the crocus stamens that are collected that end up being an important spice. Okay, so yeah, so saffron. You can see that if you have saffron, and you have to physically, people who harvest the saffron, they actually have to go and pick out the stamens from these blooming crocus plants, which are again low to the ground. So this is also very difficult, and that's why saffron is so expensive. Yeah, okay. So, and then you also have plantlets and suckers. Plantlets are basically little plants that are going to grow on the leaves of the parent plant. Kalanchoe plants, which is a very pretty flowering plant, often sold in florist shops and things like that. They grow by plantlets, and then you have suckers, right? Suckers are produced by roots. Many grasses and trees grow through these suckers. So you can have a whole grove of trees that you think these are each individual organisms, but they're actually the same organism. Okay, they're all connected underground by these horizontal shoots, suckers. Aspen trees are like this. So you could see a whole grove of aspen trees that that are basically the same organism. And I show you some pictures here. So this is again from your book, but you can definitely see the stolons in the strawberry plant. And here's the garlic bulb, and you could see that each one of those cloves have germinated. So as I said, they'll each grow into their own garlic plant. And here's an example of plantlets. Okay. But again, reproducing asexually is going to limit genetic diversity. Okay. And that can have severe impacts on the ability of a population to survive. Prime example of this is the English elm. So English elms were actually brought to England by Romans a few thousand years ago. And basically, all English elms were clones, or the vast majority of them at least, right? Were clones of each other. Very little genetic diversity. Then you have Dutch elm disease, which came from the northern continent of Europe and made its way to England and was able to infect English elms, and no genetic diversity, wiped them out, just completely wiped them out. So you see genetic diversity, how important a role it can play. Interestingly, we use asexual reproduction a lot in agriculture. For several different reasons. One being that when we have a particular varietal, a particular hybrid, a particular strain of a plant that does what we want very, very well, right? Then we don't necessarily want it to produce sexually to increase diversity. We want to sort of keep it the way that it is because of some specific benefit. Think about those strawberries, right? We like big strawberries, juicy strawberries, sweet strawberries, that nice red color. So all the strawberries being cloned, all the strawberry plants being clones, producing those big, juicy, red strawberries, that's what's going to sell. So we don't necessarily want a lot of genetic diversity kind of happening with sexual reproduction where we could lose that, okay? So we have also found other ways to create asexual reproduction. One is stem cuttings, right? So you cut a plant. A lot of times you basically can treat the cut end of that plant with some plant hormones, and then you stick it in the ground, and the plant hormones that you treat it with are actually going to stimulate root development, and new roots will form, and boom, you now have two plants that are genetically the same, that are growing. This happens a lot in flowering plants that we actually use for flowers, that we grow for flowers. Okay, so we do this. Woody plants can be propagated by grafting, where we actually we cut the top part of a plant that's doing well off, leaving the roots and the stem. And then we cut the plant that we want, and we basically take the top half of the plant we want and put it on the bottom half of the plant that's doing well. And we bind it together, and basically, some amount of time, that plant is those two plants are going to heal themselves and form one plant. The root part is called the stock. The top part that we're moving on is called the scion. Okay. This has become incredibly important in wine grape growing, particularly in Europe. There is a fungus that attacks the roots of many different varieties of wine grapes, and will wipe out a whole vineyard. And so you can imagine the economic impacts of this for a vintner, a farmer who grows wine grapes, or for an entire region. Let's say the whole area. Think about the Sonoma Valley in California, which produces a lot of wine grapes, right? This could devastate a whole community economically. So what they do is they graft the grape that they want to grow, the top part, the scion, onto root stock that is actually resistant to the particular fungus that infects these grape roots. And so then you're growing the grape variety that you want for the wine that you want, but its root stock is resistant to the fungus. And this happens on a massive industrialized scale in growing these wine grapes. Okay, let's talk a little bit about apomixis. I know we're getting short on time, but we have just a couple of slides. Apomixis is asexual reproduction of seeds. Okay, so a couple different scenarios where this can happen. You can have the megasporocyte that basically meiosis gets fouled up, and so it produces a diploid egg cell that then basically goes on and forms its own seed. Okay, you can also have diploid cells from the integument around that embryo sac, basically form their own diploid seed. Okay, this happens a lot in some species of agriculturally important crops like grasses. Many grasses do this. Kentucky bluegrass does this. Okay, some corn hybrids, this sort of happens there. So it can be important again in agriculture. There's a lot of work trying to understand how to control this, what the genes are, things like that, because you will want to control this if you can for specific, particularly in agriculture, specific plant products that you want to be able to reproduce asexually and keep that the way that you want. Okay, and there are ways. There's a lot of work trying to figure out how to do this artificially. GMO crops, right? So this is a huge market. now. A lot of the GMO crops have to do with resistance to pesticides and herbicides. So this way, the plant is not going to be affected, and you can basically douse the entire field with a lot of this herbicide or plant or pesticide, kill off the pest or kill off the weeds that are affecting your crop plant, so that you can maximize yield from a field. Trouble is, the plants that you're trying to kill, or the pests you're trying to kill, typically become resistant usually within a decade. So this becomes sort of a merry-go-round of resistance and development. There's lots of ways to introduce different DNA into plants. Some of them are really interesting. You can use viruses that will transfer plasmids in some small percentage. You can try directly ingesting the DNA, and what I thought you guys might get a kick out of, the gene gun. And it's exactly what it sounds like. It is a gun that shoots DNA into the leaf of a plant. And here you are. That's what it looks like. Shooting, you load the DNA in, and it actually shoots it at very high speed, so that it would actually puncture into a plant cell and be incorporated into that plant cell, which is wild, and it sounds like science fiction, but it's true, and it happens all the time. So there you go. That is today's lecture. I hope it was helpful. I hope you found some interesting things. Okay, so we're at 1:30. So if you've got to go, please feel free to do so. But if you have questions, hang around and I'll try and answer them. Questions, guys?
Yeah, Professor, I had a quick question on one of the diagrams you were showing for long day and short day plants.
Yeah.
There was a red line kind of in the diagram between the light and the night section. So I was wondering what that red line represents.
Yeah, so for different species of plants, there's going to be some critical value. So that when you cross that, and that's what that represents, the critical value. So that when you cross that critical value in the ratio of light to dark each day, that's what's going to be the trigger, for either producing flowers or stop producing flowers.
Got it. So when it reaches nighttime at that critical value of whatever the day length is, that's when the trigger is.
Yeah, exactly.
Okay, got it, got it. Thank you.
You got it. Other questions?
Professor, I have a
Yeah.
kind of a weird question, but like I'm kind of interested about it now from the gene gun.
Wait, okay, you got to tell me who you are because it's not popping up.
Oh, Sean Jacobs, sorry.
Oh, Sean, okay, go ahead.
Could you essentially shoot genes from the gene gun into like a human to like give them like other genes?
Yeah, I know, right? So it becomes a science fiction movie.
Yeah, exactly.
You could. Now, here's the thing, right? So it's what can you shoot in? There are also mechanisms in animals to not take up genes very easily, right? Exterior genes very easily. So different species have different mechanisms to sort of protect their genomes. Plants do it a certain way. So, you're also preparing the DNA that you're loading in there to sort of maximize what it can and can't do. But I agree with you. This is a fantastic, sort of science gone wrong sci-fi movie kind of a thing. But yeah.
All right, thank you.
Yeah, no problem. I know it's super weird, right?
Yeah, I was like, I didn't know if I wanted to ask, but then I was like, let's see what happens.
Right, no, I know. But mostly this is done in plants. Part of the reason too is because, and you saw in the photo that they were shooting a leaf, right? So the leaf has this relatively thin, high surface area kind of a pattern so that you can get deeper into the tissue, proportion wise, by shooting it into a thin layer. Yeah. So, but I mean, theoretically, Sean, it's possible, right? I mean, this is the mechanism that they're kind of doing, and I want to be very careful here, right? So please listen to everything that I'm about to say. This is sort of the mechanism that is being used in these mRNA vaccines for SARS coronavirus 2. Now, they are not injecting genes into human cells. That's not what the vaccines are doing. They're injecting messenger RNA. And basically, so it has nothing to do with the human genome at all, right? But it is putting in messenger RNA that then goes to human ribosomes in your cells, and you produce the protein, which is a piece of the spike protein of the coronavirus, that your immune system then learns, hey, this doesn't belong here, and trains itself. And that's how these mRNA vaccines are training your immune system to protect you from COVID-19 disease.
That's for all vaccines, right?
That's for all what? I'm sorry?
Do all vaccines, act the same way?
No.
No?
No. As a matter of fact, the only two vaccines that we have developed are the two just for the coronavirus. Most vaccines, what they do is they use a weakened
virus?
A weakened form of a virus or the pathogen, or they take a weakened virus and then they take a small gene from that and they put it into another virus that doesn't do very well in humans. And this is what they do with flu vaccines, basically, right? They take the flu vaccine, they get a weakened form of it, then they basically sort of create, they take, let's say, a monkey virus that doesn't do well in humans, and even when it does cross into humans, doesn't cause serious disease, right? Maybe you just feel that day or two, right? And they attach it to that virus so that that virus then gets recognized, and the components that come from the flu virus are recognized by your immune system. And so your immune system is primed and trained to knock out flu if you happen to get exposed to it before it gets you sick.
I have a question regarding what you just said. It's like off topic, but not so off topic.
Yeah.
How exactly, like before releasing a vaccine, would they test the long-term effects that it can cause?
So, would you mind repeating that? It was a little choppy.
So how exactly would they be able to test like the long-term effects that a vaccine may cause before releasing it to the public?
So typically what you have to do, well, first what they do is they test it in the lab. Then they test it on a series of different animals that are lab animals that are more and more sort of closely related to the immune system of humans. So you may start off with mice, and then you may go to pigs, and then, then it may be approved for human clinical trials, or they may want something from monkeys or even apes. They would do that, and then they test it in a group of humans. And the number of humans who have to volunteer, right? They don't force you to take it in a trial. That volunteer have to be sufficient so that the chances are good of you finding any sort of issue that comes up within that number of people. So typically you're talking about tens of thousands of people that take it. Now, as far as long-term effects,
Yeah, because I feel like that only covers the short-term effects.
Yes, but most vaccines don't necessarily have very long-term effects outside of the immune system, right? Because the thing that is being injected into you, your body's going to actually clear it and remove it in relatively short order. Where there have been long-term effects from vaccines have had not so much to do with the vaccine, but things that we added to the vaccine for other
Mercury,
reasons.
right?
I'm sorry?
Mercury, right?
Yes. So things like mercury, thimerosal, these were things that were added to vaccines to help with storage of the vaccine,
Gotcha.
storage and transport of the vaccine. And that can have a hard, a long-term effect that's difficult on those people receiving the vaccines.
Okay, fine. That makes sense.
Mm-hmm. Yeah.
I have one more question regarding something that we learned in lecture.
Yeah.
So, would rice be considered a short day plant, or is it more of like a kind of an iffy crop to categorize?
Yeah, I don't know off the top of my head. I saw that. Was it you that asked that question in the
Yeah,
chat?
yeah.
I saw it. But off the top of my head, I don't know. It may not be necessarily triggered by light because don't forget, rice is, no, I guess it could be. But anyway, rice grows in tropical sort of climates, right? So it's got to be
Mm-hmm.
pretty warm. You can't
Yeah,
can't
yeah.
really grow rice in northern Europe, or Canada. Like you're not getting a lot of rice production in Canada. We do get a lot of rice production in the Carolinas here in the States. So, and the Carolinas do clearly have seasons. So, it's more of just like a general rule, not a hard and fast rule. But I don't know enough about rice to answer your question.
Okay, fine. It's all good. I'll do some research
Yeah.
regarding it.
Yeah, no problem. Okay. Any other questions, guys?
Professor, I have a question. I have a mini question. You are available for office hours today?
I'm sorry, say that again. Guys, when you hear me, am I this choppy or is it just on the receiving end for me? I hope I'm not this choppy. Sometimes it's hard to get the questions. I apologize.
No, you're good. She was asking if you're available for office hours today.
Yeah, right. My office hours, I have my regular office hours today.
Okay, thank you.
Yeah, no problem. I'm sorry I didn't, that I'm asking you guys to repeat questions. I apologize. But yeah, I've got my regular office hours today. The other thing that I wanted to mention that I forgot in the housekeeping was, we had trouble with iClicker for our last session when I asked those several questions. I just deleted the whole session. So many of you have emailed me. Don't worry about it. It's if I can't completely if I feel that there were a lot of errors or something like that, it just makes more sense to get rid of the session. So I did that last time.
Oh, okay. Thank you.
Mm-hmm.
So starting Tuesday, it's Professor Arnott.
Yeah, who will be lecturing and holding office hours? Yes.
Do you know if he runs your class the same way, like with he posts the slides before and?
Yeah, I don't know how he'll do it. I suspect that he will post before. I don't know what format he'll do to post, but he and I are actually going to touch base to try and figure out the best way to ease the transition for you guys.
Yeah.
So we're meeting later this week. So I'll mention that you guys were asking about it.
So we still have you on Thursday, or you said he starts
Yes.
on? Oh, okay,
Nope.
perfect.
Thursday's our last hurrah.
Sounds good. Sounds good. Thank you.
Okay.
Thank you. Have a good day.
Yep, you got it.
Thank you, Professor.
You got it, guys. Okay, so I'm going to stop the recording.